Thanks everybody for being here. I have the honor to present some of our analyses on the element data through the lens of a machine learning approach as well as human written heuristics in DeepVariant. So first off, a brief introduction to the team. So our team is the Genomics team in Google AI. Our mission is to maximize the utility of genomic data. We build methods across the range of the sequencing life cycle. We have a strong open source focus. So here are 4 of our open source repositories. Our general goal is to build these methods and make them maximally available to the community. And today I'll be using DeepVariant as the open source method which does variant detection. So in DeepVariant, we use a deep neural net to take representations of genomic input data, sort of as a composite representation from the genome browser, pass it through this network, and it generates probabilities for homozygous reference, heterozygous, homozygous variant. There are a couple stages of DeepVariant. In the first component, there are human-written heuristics, which structure the input data and identify candidates. After those candidates are generated and annotated with things relevant for classification, a neural network looks into those pile-ups and creates a classification of these probabilities. And it's interesting that we have the ability to look at element data through 2 different lenses here. With the neural network, we can look into how much is learnable in the data. Because this neural network learns without the prior biases, it discovers as much of the things related to variant calling as it can on its own. And there's also the candidate generation stage, which represents how humans would write things like SAM-tools pileup or G8DK, how they approach calling in general. And so each of these are informative for how we would analyze and look at the quality of element data. So for the first thing that we do is we just take standard Illumina workflows. We're going to take BWA mem, map it to the standard GRCH38 reference, and we're going to use the DeepVariant Illumina model out of the box. And we're going to benchmark all of these things with the Genome in a Bottle v4.2.1 truth set. Oh, and to clarify, we use HE003, which is withheld from all training in this case. So the neural network has never been trained on this data. The other thing that we do is we train a model using the element data itself. So what you can see here at the top, these are the input channels. This is the actual data that DeepVariant sees. It represents read bases, base qualities, mapping qualities, and so on. What you can do is you can take the element input data, associate it with true labels from Genome in a Bottle, because you know what the positions are. You train a new model from your old one, and then use that to assess the calls. So first off, we got, I think in this case, we got HG002, trained a model. We held out HG005 and did our evaluation on the HG005 holdout. So what you're seeing here in the 3 curves are the number of errors as you downsample in coverage from 30-fold down to 15-fold. And in yellow is a NovaSeq run at the same number of reads with Illumina model. In red is the Illumina DeepVariant model with the element data, and in blue is the retrained model. So the key takeaways here are when you get to higher coverage, like 30-fold coverage, actually it looks pretty similar. It's virtually indistinguishable in quality. I mean, you can just use the Illumina models and it works just fine. When you go to lower coverages, and even in the Illumina model, in our observations, we have a higher accuracy in the element data than we do with the Illumina data. And this is a little bit further enhanced by retraining. But it's quite interesting. You don't need retraining to actually see this difference. OK, so in that part, we've asked the neural net to basically learn as much as you can about this data. We can look into that human-written component to see if we can extract some of the reason as to why there might be a difference that we're seeing in those 2 runs. So here we're going to investigate the make examples component. So one of the things we noticed right away is that the element data is producing cleaner candidates. So this is at 15-fold coverage. You can ask, of the things that human-written logic proposed, how many of them were rejected by the neural network? How many of them were eventually called as variant? As you can see, in Illumina, the neural network has more of a job to filter because there are more of these candidate sites that are proposed. And in general, the proportion increases as the coverage goes down. And we don't see that same phenomena in 15x for the element data. And interestingly, when we go to 30-fold coverage, we also see it's a reduced observation of the phenomenon, but we do see the same phenomenon. And what this indicates is that there is some additional kind of cleanness or noise reduction that is not as apparent after you've gone to like full high coverage human genome sequencing and then called with a neural network. But there's something in the underlying data which does actually appear to have an advantage. So we took, Element had given us a number of runs. We've also generated Illumina data from a lot of runs. And we just asked that same proportion of candidates that are rejected. I think this was at 18-fold coverage for all of these runs. And we do see that this seems to be a consistent phenomenon. I'll point out you do see also minor differences between the Illumina platforms as well. So you can see some platform-to-platform variation. It's not clear if this platform variation actually matters for the downstream variant calling because of the further refinement in neural network. Finally, I wanted to talk about the trio calling analysis that we've done. So our team has developed a method called DeepTrio, and it leverages the fact that the powerful component of DeepVariant is very easy to retrain. You take input data, structure it, give it labels, and get results. And for trios, you can actually do this very interesting phenomenon where you normally take one pile-up, and we had the idea of like, let's just expand it. Let's vertically just take 3 pile-ups, and we'll literally just composite them in the same image. And so what this diagram is showing is this is the input data for child, mom, and dad, and then all of the different input channels. So this is now what the neural network sees. And then we have a parent model which classifies the parent, and a child model which classifies the child. So we received one trio to train a DeepTrio model and generate calls both for a genome in a bottle set as well as for additional trios. And we now take chromosome 20 because we have only one trio to train on. So we're going to hold out chromosome 20 and train on the rest. And one of the things that we observed is a smaller number of false de novo calls. I should point out these are cell lines, so the number of de novos is actually going to be higher than in an individual. But because we have genome in a bottle, we can annotate which of those de novos are true and which ones are false. And we do also observe lower, at least in this trio, a lower false de novo call rate, which makes classifying rare disease substantially easier. So finally, in summary, for typical whole genome coverage, Element is clearly able to match state-of-the-art with standard, even kind of currently existing workflows without any modification, at least in the ones that I've looked at. There's existing short-read software. It works well out of the box. When you go to low sequence depth, there does seem to be this cleanness in the error profile, which might enable other applications. And I think it's an interesting question. It's maybe less relevant for deep coverage variant calling, but for things like somatic detection, or cell-free DNA, or maybe like low-pass calling and imputation, are there applications that can take advantage of that apparent additional signature? Finally, we do have retrained models that will improve performance, and we anticipate making all of these available as open source and available in the same standard way that we do for our other DeepVariant models. And finally, I want to make a special shout out. Sention has optimized variant calling models as well, and also has an optimized mapping approach, and they've been working with Element closely. I think they have an application note. You should feel free to check it out, and we have no concern if you use Sention software either. It's all great. We just want to see the field move forward. And finally, thank you from the team. I've highlighted the group of folks here who work on DeepVariant. We also have a number of those other applications that I talked about early on. And thank you all, and I'll now hand it off.